Glass sponges, as the name suggests, have a glass-based skeleton composed of a network of glass needles, hooks, stars, and spheres. To achieve such a unique architecture, they have to manipulate the shape of disordered glass to form highly regular and symmetrical elements. Thin crystalline fibers made of a protein, it is known that silicatein crystals are responsible for glass synthesis in sponges and for shaping the glass skeleton. However, until now efforts to determine the 3D structure of this protein, describe how it assembles into crystals, and how those form the glass skeleton were unsuccessful. Mainly, because nobody was able to reproduce these crystals in the lab. A team of researchers led by Dr. Igor Zlotnikov from the B-Cube Center for Molecular Bioengineering at TU Dresden tried an unusual approach. The Zlotnikov group, together with researchers from the Dresden Center for Nanoanalysis, DCN, at the Center for Advancing Electronics Dresden, CFAD, used high-resolution transmission electron microscopy, TEM, to take a closer look at silicatein crystals packed inside the glass needles. We have observed an exceptionally ordered and at the same time complex structure. Analyzing the sample, we have seen that it is a mixture of an organic and inorganic matter. Meaning that both proteins and glass form a hybrid superstructure that somehow shapes the skeleton of sponges, explains Dr. Zlotnikov. A traditional way of obtaining a 3D structure of a protein is to expose its crystal to a beam of X-rays. Each protein crystal scatters the X-rays in a different way, providing a unique snapshot of its internal arrangement. By rotating the crystal and collecting such snapshots from many angles, the researchers can use Such an approach is widely used and is the basis of modern structural biology. It works well for crystals of at least 10 microns in size. However, the Zlotnikov group wanted to analyze silicatein crystals which were about 10 times smaller. When exposed to X-rays, they were almost immediately damaged, making it impossible to collect a complete data set of snapshots from multiple angles. With support from the team at PSI's Swiss Light Source, SLS, the researchers used a new emerging method known as series. With the new method you get many snapshots which you combine afterwards to decipher the structure. Each snapshot is taken at a different part of the tiny crystal or even from a different crystal. In total, the researchers collected more than 3,500 individual X-ray diffraction snapshots from 90 glass needles at completely random orientations. Using state-of-the-art computational methods, they were able to find order within the chaos and assemble the data to determine the first complete 3D structure of silicatein. Using the newly obtained 3D structure of silicatein, the researchers were able to understand its assembly and function inside the glass skeleton of the sponge. They built a computational model of the superstructure within the glass needle and explained the initial complex images of the protein glass superstructures obtained with that atom. In fact, what we describe is the first known naturally occurring hybrid mineral-protein crystalline assembly, concludes Dr. Zlotnikov. Story source, materials provided by Technische Universität Dresden. Note, content may be edited for style and length. Please support our channel to grow by pressing subscribe button as well as the bell icon for daily science updates. Thank you.